So next is components of regenerative endodontics. Hargreaves et al. Have identified three components contributing to the success of this procedure. They include three S. First one is system cells that are capable of hard tissue formation. Second one, signaling molecules for cellular stimulation, proliferation, and differentiation. And finally, a three-dimensional physical scaffold that can support cell growth and differentiation. So, components of regenerative endodontics is scaffold, growth factors, and stem cells. Stem cells. Stem cells are undifferentiated embryonic or adult cells that are capable of differentiating into various specialized cell types. Stem cells may be pluripotency, that is, plus self renewal. The fundamental property of stem cells is the ability to go through numerous cycles of cell division while maintaining the undifferentiated state. In addition, stem cells produce intermediate cell types called progenitor or precursor cells that have the capacity to differentiate into different cell types and generate complex tissues and organs. Differentiation or occurs when a stem cell acquires the features of a specialized cell. Stem cells can be either embryonic or adult, that is, adult is postnatal. Embryonic stem cells are isolated from the blastocysts during embryonic development and give rise to the three primary germ layers ectoderm, endoderm, and mesoderm. These cells are totipotent or pluripotent with an unlimited capacity to differentiate and can develop into each of the more than 200 cell types of the adult body. Adult stem cells exist throughout the body in different tissues, including bone marrow, brain, blood vessels, liver, skin, retina, pancreas, peripheral blood, muscles, adipose tissue, and dental tissues. They are localized to specific niches where the regulation of stem cells, proliferation, survival, migration, fate, and aging occurs. An adult stem cell can divide and create another cells like itself, and also a cell more differentiated than itself, but the capacity for differentiation into other cell types is limited. This capacity is described as being multipotent and is a distinguishing feature of adult stem cells compared with pluripotency of embryonic stem cells. So stem cells can be basically divided into pluripotent and multipotent. Pluripotent is found in developing an embryo and multipotent is found in adults. Types of stem cells based on the potential. Stem cell type, totipotent. Description is each cell can develop into a new individual. Examples, cells from early 1 to 3 days of embryo. Purely, pluripotent cells can form any over 200 cell types. Some cells of blastocysts 5 to 14 days. Multipotent cells differentiate but can form a number of other tissues, fetal tissue, cord blood, and adult stem cells. A tooth develop as a result of carefully orchestrated interactions between the oral epithelial ectodermal cells that form the enamel organ and cranial neural crust derived mesenchymal cells that form the dental papilla and dental follicle. Mesenchymal cells give rise to other components of tooth, that is dentine, pulp, cementum, and periodontal ligament. Beginning in 2000, several human dental stem or progenitor cells have been isolated and characterized. Dental stem cells. Different dental stem cells are dental follicle stem cells, dental pulp stem cells, DPSCs, stem cells from human exfoliated teeth, periodontal ligament stem cells from apical papilla, and gingival fibroblastic stem cells. Stem cells from apical papilla. Stem cells from apical papilla cells are found in apical papilla, located at the apexes of developing teeth at the junction of the apical papilla and dental pulp. The apical papilla is essential for root development. Stem cells of the apical papilla cells were first isolated in human root apical papilla collected from extracted human third molar. The cells are clonogenic, and can undergo odontoblastic or osteogenic, adipogenic, or neurogenic differentiation. Growth factors, morphogenes. Growth factors and cytokines may act as signaling molecules that modulate cell behavior by mediating intracellular communication. Growth factors are polypeptides or proteins that bind to specific receptors on surface of target cell. As opposite to systemic effects by hormones on target cells, growth factors or cytokines typically act locally on target cells. Among the growth factors released from dendrin matrix, TGF beta 1, fibroblast growth factor 2, and platelet derived growth factors enhance cell migration. PDGF and vascular endothelial growth factor control angiogenesis. TGF beta 1, FGF F2, VEGF, and insulin like growth factor stimulate cell proliferation. Bone morphogenic proteins and FGF2 promote dendrinogenesis. Non collagenous proteins such as dendrin matrix proteins and dendrin phosphoprotein and glycosaminoglycans such as chondroitin sulfate and dermatan sulfate also promote dendrinogenesis. Platelet derived growth factor.